Okay. So I know that this isn't technically lab, and I know that in lab we are not doing the gram stains today, but we will be doing gram stains on Wednesday. And I want to be sure that everybody is going to be ready to do them. I will not be helping you on Wednesday. Whatever answer you guys get for which one is your gram positive and your gram negative carries with you for the rest of your semester. It doesn't have the same number as what it did the other time. So if you get a different answer, and you are confused, but you have no idea how to ask the question on how to help, I'm not helping you. Okay. You need to understand the material well enough at this point in the lab for the gram stain, right? Because that's like the first stain we did. So if you don't understand the gram stain, you need to pick your shit up and start understanding the gram stain, because you should know it by now. I think we can all agree with this, right? It's the bread and butter of everything. So that's why this is up here. Gram positive, basically, let's just go over to this part of it. Yeah. So gram positive should be purple. I used to give this as like a quiz and people would like turn it in after, but I don't want to do that today. Nope. So gram positive is purple, positive purple, positive purple, P purple, negative red. To me, red is a negative color. Like I've said before, looks pink, but it is definitely not purple. If you guys remember for like questions 1 and 2, and then questions 3 and 4, questions 1 and 2, you look in the microscope and you see, this pinkish color. Some people were not sure if they thought it was purple or not. But what did you think when you saw the next one? The next one was flipping purple, right? Like there was no question about it. So if you're not sure about it, think about what you expect the purple to really look like. Like it will be purple. It will not be, maybe it's a little purple. No, okay. It'll be purple if it's positive. Pinkish is fine, but we'll call this red for the sake of argument, all right? So primary stain, crystal violet. Crystal violet is a stain that, sticks itself in between the structure, that lattice framework of the peptidoglycan wall. Iodine is our mordant, okay? Iodines are mordant. It helps stick that crystal violet in place if it did get stuck in there, right? We rinse with ethyl alcohol. That's our decolorizer. The decolorizer just rinses out any of the crystal violet that didn't get stuck in place. And then our counter stain is safranin, because if you're gram negative, none of the crystal violet got stuck in place. You rinsed it all out and now you have clear cells. So you want to be able to see your cells. That's all that the safranin is there for. Now, first question, where does the positive's color come from? Which stain? So we know positive was purple. Its color comes from crystal violet, right? Easy. All right, where does the negative's color come from? Which stain? We know the negative is red. Where does the red come from in all of this? Our counter stain is safranin, right? So it should be obvious and it should be straightforward for that. Is safranin red color present in the gram positive sample? Yes, it's just covered up by that, really bold purple. It's still there, okay? It's not like you're not staining with the safranin. It's still staining it red. It's just such a darker color that you don't see it, right? So that's all that's going on there. So that is, to me, the basics of the gram stain. That's all you needed to know for the lab exam for the gram stain. A lot of you guys are literally wasting your time reading shit you don't even need to read, honestly. And then you get to the gram stain questions, and you've got all this information crammed in your head that you never needed. And it's like you get to the questions and you're just like, your brain shuts down. It's just like too much for it to like sort through or something, with the anxiety, with the stress and everything like that. Y'all can't tell me that. So many people come up to me after the lab exam, right? And they're like, man, that was way easier. I feel so much better about it. Were your grades reflective of that? Not really. Right? I don't know if you guys know this, but the average was like 60. Okay. So it's the same as the other tests were. I think you guys just was the highest. I think you guys got a 67 average, but everybody else's was closer to 60. So yeah, legitimately you guys did the best on it, like no joke. So but yeah, so what happens though? What happens there? And it's just like the lecture test. We've talked about this before, where you understand what I'm talking about in the lecture. You understand what you're doing in the lab. You understand when you're reading through it. It's like it makes sense when you're reading through it. But somewhere in there, when you go to take the test, all of that information just seizes up in your brain with that anxiety and the test taking and everything like that. and it just doesn't translate in. So you know that you knew the information and you took the test, so you must have done well, right? Because you understood what the step is. If I go over those questions with you in lab, and I'm happy to do it, if I go over those questions with you in lab and go over each question again, it's going to feel just like the lecture test stuff. Dude, you're going to be looking at the questions that you missed and being like, geez, how did I get it wrong? You know, that's what it's going to be like. I'm happy to go over it. When we will do so after we, talk about the unknown report, which is nothing, but I just wanted to let you guys know that, we're not done with this stuff, right? You took the midterm exam. Is that stuff on the midterm exam gone? Absolutely not. You're going to see all of that again on the final. Okay, not the same questions, but the same content. So you can't just let it go. That's why I'm bringing that up. How do people do usually on the final compared to the midterm? About the same. So bank on that. Okay, I'm telling you that now. If you need to, if that means stepping up on your homework or your quizzes or something else to keep that grade up, that I'm just letting you know that test is not going to bring your grade up. The final on the lab exam anyways. Okay. People do better typically on unit 3 than they did on unit 2. Obviously they did better on unit 2 than they did on unit 1. And it tends to progress that way. But the problem is unit 4 doesn't follow. That's because unit 4 is comprehensive. So it's a lot more information and it's 100 questions. It's a final, So we've got all of unit 4 stuff and the final stuff. So it tends to be more of an average of what people have been doing on the tests. So if y'all are sitting there thinking that you're going to get some kind of test that's going to bring your grade up, no. Okay, it's not going to change, right? And to me, and I think if we were to go over the questions and talk about the questions and everything like that, and you could tell me that you understand the material, you didn't do well on these questions, but you think that the questions are fair, at least with regard to the study sheet, which is me babying you in case you didn't know that, y'all. That's babying, okay? If that isn't enough for you to pass this class, I will not pass you, okay? To me, that's babying. If you can't make it through with the babying, the easiest microbiology course in the state right here. If you can't make it through that, I'm not going to pass you. That's how this goes. Okay. So that's why I say, look out for your test grades. If you think that you've been, well, I've been studying, I cram, the night before and I do pretty well. Maybe if you think that's good enough, then that's fine. If that's going to get you a C and pass you, that's fine. But if you're on the line, the tests aren't going to bring you up, y'all. It's not. Okay. The tests are only going to bring you down and they're worth a lot of your grade. All of the tests altogether are about 35% of your grade. It's a lot, right? And we all know we have the quizzes that people are pretty lazy on those as well. I'm just saying, just because people are doing poorly on them and you have access to all of the answers while you're taking them, I don't understand how people are getting questions wrong on these, but it is what it is. That's how you choose to answer them, and that's how you choose to answer them. I can't help you there, but that's all I'm saying. Right. So I have told other classes. I don't know if I told you guys. I don't think I have, but I think I may have in the lab. But I'll say it again, which is that when I was in graduate school, right, or even in, let's say when I was in undergrad, and I've talked to Dr. Tim about this. When I was in undergrad, my classes, especially microbiology, when I got to this class, literally this class, when I took it, lecture and tests. That was it. There's no homework. There's no study guides. There's no study sheets, nothing. You got the lecture, you have the textbook, you take the exams. That's what college was like when I went to college. When I say I'm babying you, I'm not kidding, okay? That's what this class was like when I went there at OSU, okay? When I got to graduate school, I took a class on cell and molecular biology, which is what my major technically was in graduate school. And we had 2 exams, the midterm and the final, that was it. There were 5 questions on each exam. They were 2-page long essay questions. There was no limit. They didn't say you had to write 2 pages or anything. They asked you a question on explain the process of blah, blah, blah. And you had to write down everything that we learned in that class about that process by hand from memory. Now, what did we do to prepare for that? They gave us lectures. That's it. There's no, there was a textbook for that, but it didn't really follow what they were lecturing. Because these are experts in their field, right? And they're training people who are going to be experts in that field. They expect you to pick up your mess and figure it out, right? So that was next level. But this is somebody who made it through that and was doing very well in that scenario, who is telling you, you are not doing this right. You are not studying effectively. You're just not. If I can go to that sort of class at an Ivy League level and not go to the lectures, I didn't go to those lectures. I didn't attend them. I just watched the videos after. And it wasn't like it used to be. It didn't used to be like this, is what I'm trying to say. There was no like YouTube or anything like that when I was going to college back then. That wasn't really a thing that everybody could like post onto and like inform their videos and all that. It was not feasible really. So they had their own website that you had to go to and then watch the videos. What I did the week before the exams was with my friends, sat down and watched the lecture videos, right? We sat down and watched the lecture videos and we had our paper in hand, our notes. There was no study guide. Again, that wasn't a thing, right? No study guide, no outline, nothing like that. That's on us to figure out what's important. Sat down with my friends and we went through every single video word for word. If I didn't understand a topic, it was on me to pause it and ask my friends what's going on here. And if they didn't know, to look it up in the book or look it up somewhere where they could figure it out, right? That's what I had to work. And that's how I did well in doing that. But that's the matter of taking time to understand it, which I think is a bigger problem when I tell you guys that you need to be taking these exams and understanding the material. I don't mean you understand the words in English. I mean, like you literally, if I ask you questions about this material, can answer questions about it further than the statement on the study sheet, right? That's what I'm talking about with understanding. That's what the study group is for, okay? When we go into lab, if you don't want to stay and sit through that shit, that it's fine. I will not judge you if you think you're doing fine or your study methods are working good for you. There are a few people in here who I would believe that, okay? But there are some people in here who I think need help with that. And it's not because you are not intelligent or anything like that. That's absolutely not it. You just don't know how to study or take tests. Nobody taught you how. They gave up on you. They got lazy on you. And now you don't know how to take tests or study or anything like that because nobody took the time to teach you how. They just expected you. They just threw you into the deep end. And some classes that you go to, like you're probably like Comp 1 and shit like that, that's like not a real class. When you go to classes like that, where they give you lazy person tests, they do, right? So it's like the kind of test that's written to get you out of their class, right? So when you get to classes like this one, and like physiology especially, right? That's the two that really are going to get you. And a little bit of anatomy, but it's not quite the same. These classes are the ones that are like really getting you to that point of like, yes, I'm teaching you brand new material that you've never seen before. And I expect you to understand it and do the work to understand it. And then I'm going to test you about that. And no other, and it sounds ridiculous. It sounds simple enough to say out loud, but I bet you've never had a class that did that other than these two. I'm just guessing. Okay. If you had high school anytime in the last 10 years, I would say, then I can guarantee you don't know how to take a test, okay? Because them teachers are lazy. They are, especially over COVID. COVID was everybody just pushing it. Nobody wanted to deal with it. Just push everybody through. And so I feel like that is a lot of where, even if you were already an adult and you've already gone to, high school before, going back into college through some of that or taking some of your prereqs through some of that doesn't prepare you for classes like this. It doesn't. So that is me. That's my call to reality or whatever for the day. So this always happens around midterm. I like to kind of give people a little bit of a slap in the face for that stuff, just because I want you guys to remember it doesn't get easier from here, right? The stuff that we've been learning up to this point is prepping you for what's coming, which is harder stuff, not easier stuff. It's not like we reached the climax of the hill and now it's all downhill from here, right? So that's what I want to remind you guys about is like, if you weren't doing good up until now, like you're looking at your scores, you're like, oh, it's low. I'm sure like, as we go through this will get easier. Can you tell me about macrophages and neutrophils? Can you tell me about what, T cells and B cells do right now? Because if all that stuff sounds like, what to you, which it probably does, then this is going to be harder information than the previous ones. Okay. Most of you have heard of DNA. Y'all probably don't even know what a B cell is. If I say lymphocyte, I bet some of you will be real cheeky and, oh, yeah, I work now. I know what lymphocytes are. Do you? Tell me about it. This is one of those scenarios Or if I was meaner, I probably would be pointing fingers at people and telling you, tell me what lymphocytes are and shit like this, because you should be able to, I should be able to do that and you should be able to answer it. So if this makes you jump in your heart, like, no, then you're not studying well, literally, okay? Because you should know it that well. All right, so let's get into, I love the immune system, by the way. I know I told you guys, like, chapter 10 is my favorite chapter. It's my favorite to teach because it's so exciting to like introduce to you guys how, why you breathe and all this crap like that. The immune system is so cool. And most of the time when people get to me, they haven't learned anything about the immune system. They haven't even learned hardly anything past the fact that, you know, CBC does count neutrophils or lymphocytes. They don't know what that means or what that does for the body. So let's get into that stuff, right? So as far as how you're going to fight infection, that's what these next two chapters are really going to get into deep, okay? We have two lines of defense within this category that we call innate immunity. So innate is just the stuff that's like the basic stuff. It's not specific to any specific thing in general, but it's just going to target bad. Okay, so that's our innate system. So our first line of defense tends to be our physical barriers, like literally the skin, right? Having skin, having hair, and stuff like that. Having mucus in your sinus cavity catches the microbes, right? That's a physical barrier. And we have the microbiome. We've already talked about the microbial antagonism, how they take up the seats so that bad guys can't sit down. And then the chemical barriers with things like, yes, acid in the stomach, that definitely counts. But we're also talking about like lysozyme that can help break down peptidoglycan. That's something that we're going to learn about in this chapter. All right, so that's the basic like aspect of the body that's just set up itself for protecting you. But this really what this chapter is going to be about is more about your second line of defense. And this is going to be your cellular system that fights just bad guys in general. Okay, it doesn't target a specific bad guy like what we'll see with our acquired immune system. That's our lymphocytes, by the way. But these guys, our second line of defense, they will recognize something as just being bad guy stuff. And then they go through and they perform phagocytosis. They eat up that stuff, foreign material, what if it just looks like bad guy or if it's just trash lying around, they'll eat it. Inflammation, we'll talk about the importance of it, as well as fever. It goes kind of hand in hand with the inflammation response a little bit, and then antimicrobial products that are involved in like the bloodstream, as well as cytokines and things like that, that signal the body to call in to these guys. The good, exciting, I guess, aspect of the innate immune system, it all goes in there and acts pretty generally, but it doesn't have anything like really heavy hitting, like what we will see with acquired immunity. Acquired immunity with our lymphocytes, our B cells that make our antibodies, and our T cells that go around killing cells that are infected actively. Those are our heavy hitters, but they take a long time to generate. The innate immune system is there for you while that is building up. We'll talk about how quick that can be, but it is sort of like you get exposed to, like, let's say strep, right? And your body sees that as foreign and starts like eating up just bacteria. It's like, oh, this is bacteria, eat the bacteria, eat the bacteria, digest it down. And then it's going to go activate our lymphatic, you know, our B cells in our lymphatic system and our T cells as well. Anyways, those will be triggered to multiply and differentiate and all this, but that takes so much time. These guys are still out there eating up and trying to fight and start inflammation and induce fever and whatever. That by the time that we get antibody against that antigen, right? And by the time we get T cells that are killing our infected cells and activating other cells, when they show up, it's almost like instant how quickly your body can, you know, get over that infection. It's so effective. It's ridiculous. So all of that moment in your illness where you start feeling like crap, where you feel sore, where you feel tired, where you have a fever, that's your innate immune system. Your body is just trying to get that acquired immune response kind of going, kick-started. It just takes a while for that to happen. This guy watches the door. This is like the front lines, right? This is your Marines, basically. The guys that are at the front lines fighting off all the invaders until the specialists come in, right? Or you could even say, not even just a specialist, but the guys with the friggin' tanks. Like you could look at it that way, you know? So these guys are doing the best they can with what they've got. But what the other guys have, they have to get that stuff together before they get there. But when they get there, the war's over, you know? So that's the idea. So whole point of immunology is that system that's going to be surveilling for foreign things, marking it as foreign and destroying it, right? How do we recognize something as self versus non-self? There are markers on the surface of cells. We've already talked about some glycoproteins, or for an example, right? They could be the lock that the virus key fits into. We've talked about that previously with the virus, but they also could be anything on any marker on any surface of anything, pretty much. Okay, but the ones that we see as self versus non-self, ours look like what we're expected to look like, and then non-self looks foreign, to the point that if I take your kidney and shove it into my body is going to probably fight it off and see it as foreign. So it's that specific. Even though it's human and human, it's not that easy, right? We know you have to be tested in order to donate an organ to somebody. It's not like you can just take anybody's kidney and shove it into somebody. Otherwise, we wouldn't be having these problems with organ transplant and the need for organs. But so you have to be very closely matching. Otherwise, there are all these markers on the surface of these cells that your body could see as not yours, right? And it trains itself early on to see self and to recognize self as this is not, dangerous, but it hasn't seen your kidney before. So as far as it's concerned, you're just a bunch of, bad guys.
Does that work with synthetic organs?
So with synthetic organs, so they have been designed for the most part. They can be designed to, some of them have like a scaffolding, right? So they'll build off of the person's cells that needs the kidney. So they'll take cells from that person's stem cells basically, and they can use a scaffolding that's basically an extracellular matrix type setup and build organ from that based on just the tissue. And a lot of times they found that structure on that actually helps drive like tissue changing, like formation of valves in a heart, for example, as opposed to like the muscle tissue. But that's tricky. But either way, even if you can control something like that, other things like if we're talking about something like a pacemaker, or if we're talking about something like that, or anything, right? So the non-living material that I'm putting into somebody's body, let's even look at it that way. Those things they look like stainless steel to our body, right? And so does our body react to stainless steel? Well, a lot of the metals in stainless steel, or the aspect of stainless steel specifically, we use it for surgery and stuff like that because we don't react to it, right? You're less likely to be allergic to it and have a reaction to these things, but they don't have like the markers that all of these other cells do. We're going to introduce some of the markers in this chapter, but like it is to the point where like if anything ever needs to get in your cell, it has its own channel or mechanism of getting in, and those are the markers I'm talking about. Anything on the surface of the cell can be a marker to recognize as self or not self. So when we're talking about artificial organs or other apparatus like that, the issue really isn't so much about self versus non-self, so much as it's like they don't have the things there for us to react to a lot of the time. So we don't react. If you're getting something like a pig valve or a cow heart valve, like heart valves from animals, right? So that's a thing for those of you that don't know. You could potentially see that as foreign, but it's unlikely because usually it's just like cartilage and there's not enough going on that tissue for us. It's not actual like true cells as much as it is like just, I don't know, cartilage isn't really, I don't know how to describe this, but yeah, it's not like all of that's going on in like body tissue cells. It's just that one valve. And so you don't really need to worry about the immune system attacking it as much. It can, but it's just not as likely. Okay. Alrighty. So yeah, I think we get into that a little bit in the, not next chapter, but the chapter after that. So like organ rejection. For those of you that don't know, if I haven't said already, I used to work in the transplant lab at Integris. So I did all of the testing for transplants at Integris for a while. That was just a little too stressful for me though. But yeah, I used to use that. So I used to do that. So I know quite a bit about it. All right, so antigens. We can call them markers. We can call them immunogens, whatever you want to call them. They're just things that your cells can see and react to potentially. That's all it is. And I think a lot of you guys kind of understand that concept already. So it's usually proteins and or sugars, like glycoprotein is sugar with protein. All right, so we need to see things as foreign and not attack things that are self, like we've mentioned. Otherwise, if we attack things that are self, well, that can cause autoimmune disorders. And you can have illnesses where microbes look like self sometimes, and they're close enough that whenever you make an antibody against them, but far enough because they look foreign, so you're making an antibody against them. They're right on that line so that when you make the antibody against them, they happen to also react to you. So that can cause things like rheumatic fever and stuff like that associated with like strep throat, okay? And that can lead, they think potentially that sort of thing is what leads to the development of other autoimmune diseases like rheumatoid arthritis and other things. We think it may be related to not just malfunction of your immune system, but potentially where things went wrong with some other previous infection of some kind. We'll get into that later. Okay, so we have these molecular patterns. This is definitely on the test, by the way. I know PAMPs. You're going to need to know what PAMP stands for. These are pathogen-associated molecular patterns. Pathogen-associated molecular patterns. These are any sort of just general pattern on the surface of a cell or as part of a cell that our body instantly sees as pathogen. That's all. Okay. So a lot of microbes are going to have these in common. So our body, if it sees those and interacts with those and says, this is a PAMP, then it kicks into gear with our innate system, right? So they are recognized by pattern recognition receptors, which I won't ask you about, but that's what they're recognized by. So they have a receptor they interact with. All right, next we have this concept of the mononuclear phagocyte system, or the MPS. We have extracellular fluid and spaces around the tissue, which is the interstitium. That's a space contained. That's going to contain that fluid. We have the bloodstream. We have the lymphatic system. We're going to get into all of these here in just a moment and what role they have in all of our immune system. So the lymphatic system itself, it says auxiliary route for extracellular fluid to go back to the circulatory system. So that's one general way of looking at it. So any fluid that is not like literal blood, right, can go back into our circulatory system and be recombined into our whole of our liquids, basically fluids, via the lymphatic system. So that's essentially what it is. So it's like a drain off system for the tissues in general, but especially for the inflammatory response. And you've probably always got some sort of inflammatory response going on somewhere in your body. If you stubbed your toe yesterday, then you probably got an inflammatory response going on there. Even just simple stuff like that, you know, bump your elbow and that sort of stuff. Inflamed from maybe injury, could be inflamed from allergies, anything like that. So if your body is responding in an inflammatory way, there are special cells and special fluid associated with those cells, and all of these signals coming to those areas, they all get transported by fluid. And so to get the fluid so it doesn't build up too much in those areas, we need a way to get it back, and that is kind of what the lymphatic system does for us. It gets the fluid back, as well as the inflammatory response dies down, the cells can come back that way too. So that's what the lymphatic system does for us. It also is a key player in surveillance, recognition, and protection against anything foreign. Because in our lymphatic system, we have our lymphocytes, a whole bunch of phagocytes, and the production of antibodies. So lymph is plasma-like fluid carried by lymphatic circulation, formed when blood components move out of the blood vessels and diffuse and migrate into the lymphatic capillary. So basically the fluid's coming back, okay? It'll transport white blood cells, fats, cellular debris, and infectious agents. If it came out of those cellular spaces into this area and gets washed through in the lymphatic system, it's going to go through this whole, it's like think of it as like a slide ride or something like that. Once you're on it, man. That's it. So, okay, these are the lymphatic vessels are going to have very thin walls, and basically extracellular fluid, that's what ECF is here, can transport across those cell walls. And that's how it kind of migrates in and out of the lymphatic system. So it's not like an open system. Neither is your bloodstream, by the way. A lot of stuff goes in through the cell walls of your blood vessels or between the cells of your blood vessels, as opposed to it like being open. All right, lymph, the most important thing that I can tell you about lymph, it only moves from the extremities to the heart, okay? So we know that blood flows out from the heart, goes out to the extremities, and then comes back from the extremities back to the heart, right? It's oxygenated on its way out typically, and then deoxygenated on its way back typically, right? So that's the idea. The deoxygenated direction is basically the way that our lymph is going to flow. That's just because if you have inflammation out there and you're trying to come back, then that's coming in and heading towards the heart, right, to get recirculated in. But that's the idea. So lymph moves in only one direction from the extremities back towards the heart. It also is only transported through the contraction of skeletal muscles. Now, I'm already telling you guys that if you have inflammation in an area, and that inflammation is associated with your immune response, and there's a lot of fluid being, moved around as a result of all of these things. And it's important to move that fluid because if you have your injured toe and it's got a lot of fluid building up as cells come in there to do their inflammatory thing, you have to get rid of the fluid and the cells as they build up, because otherwise you just got a whole buildup of fluid that can't get out of that area, right? So that's the whole important aspect of that cycling through the lymph system there. So you guys probably know this more as swelling, and you probably know this more as edema, right? So if you've ever been in a hospital and you have had patients that they are encouraging to walk around because, after surgery, it'll help you heal. Why do you think that is? Because you need your lymphatic system to move, you need your lymph flowing to help with the swelling, but you also need to get your white blood cells moving better. Because like I just told you, your lymphocytes and your antibodies are in your lymph, right? So if you want to get like healing aspects of those fighting off any potential infections, you need to pump things through those areas. And if they're not going to pump by themselves, you have to walk around to move it. If you lay there, it just sits there, okay? So you are sick, find yourself sick with COVID, flu, whatever it is, and you are able to walk around, you don't have 105-degree fever, right? It's reasonable, you got like 102-degree fever and you can walk around, try to get up like every hour and just walk around the house a little bit to get that lymph moving. It'll help your body fight off the infection a lot faster, okay? Okay, so only moved through contraction, there it is, of the skeletal muscles. If we were to compare the circulatory system, which we're very familiar with the lymphatic system, they often line up specifically because of their interaction with the circulatory system. I'm not going to ask you to point out all of this stuff in here, but it's the same concept here. Lymphatic ducts carrying from the extremities through lymph nodes. This is an example of the lymph nodes in the groin. I'm sure you guys are familiar that we have lymph nodes in the groin. We have pretty prominent ones in the groin and in the underarm and like the neck here, right? So you can palpate those whenever they are getting pretty swollen. There's other ones you can probably find in your body. You have quite a lot of lymphatic tissue in your gut. And that has to do with your microbiome in your gut training your body what is friendly and what's not. So. All right, so I think we're already learning some stuff about the immune system you probably didn't know. So the organs that are involved in the lymphatic system, we have red bone marrow and the thymus as our primary lymphatic organs. This is where our cells are going to be coming from originally, like our stem cells for all of this, as well as where maturation of these cells can often occur. Then we have these secondary lymphatic organs. This is where you have to, like the naive cells getting like triggered and activated typically. When we are going to respond. So the lymph nodes, the spleen, salt, malt, and galt. So salt is going to be skin associated. M is mucosa associated. Mg is gut associated. There's all sorts of like lymphatic tissue all throughout the body that is associated with specific aspects of the body. So that's what that's talking about basically. It's different from the lymph nodes. It's just tissue that does the same thing as a lymph node, but it's just tissue that's not in a lymph node shape. Let me tell you about a lymph node, y'all. Being from the transplant immunology lab. If you guys took a pea, like actually a green pea, and cut it in half, that half of a pea is about the size of a healthy lymph node. That's how they should be, okay? That's the size that a normal one is. Just trust me. And even like whenever we would get lymph nodes in from donors who like were maybe not all like, completely well, theirs weren't very much bigger than like maybe 2 or 3 peas. Ever. I never got anything bigger really than that. Because if they were really infected with something, then we wouldn't probably want their organ, right? That's the idea. And if they were smokers, I could tell they were smokers because in the lymph, because of the filtering aspect of your body, they had black lymph nodes. So yeah, you could tell, man. And they would be like crunchy too when you cut them up. But yeah, we would have to cut them open and like flush them out with fluid to try to get all of the lymph cells, the lymphatic cells out of there. But yeah, lymph nodes are supposed to be really tiny. So when you feel them, you know, like there's a bunch of activity really going on in there. And it's typically going to be associated with responding to an antigen, right? So, all right, red bone marrow, we kind of know where this is. It's going to be in your long bones. And it's where our B and T cell precursors are going to come from. And a lot of our blood cells originate there too. The thymus is where the T cells come from. So after they come from precursors from the bone marrow, move to the thymus and then mature there. So this is only the T cells. Okay. There are lymphocytes. There's two kinds of lymphocytes. There's T cells and B cells, and that's it. All right, lymph nodes are, like I said, bean shaped, and they're pretty much bean sized, if even. And then we have loose aggregations of tissue that we have in those alt areas, but it still has the same function. It's just not shaped in the bean shape. And they have this makeup of what's going on in each area. So there's little backflow valves and cortex and all this. I don't know. I'm not going to ask you about it, but they have this big setup of basically what's going on in each area of the lymph node. We know enlargement of lymph nodes reflects replication of the lymphocytes during immune response. And a lot of times you will have immune response going on that you wouldn't even know was going on. Generalized lymph node enlargement can indicate the presence of systemic illness. So it's saying if you get lymph nodes enlarged kind of all over general in the body, probably systemic illness. If you have lymph nodes that are localized, like let's say you had lymph nodes only like on this like underarm area, then maybe you have an infection in, I don't know, milk ducts, or I don't know, you have a cut or something on the arm, something like that would be localized infection. The spleen serves as a filter for blood instead of lymph. So it's not receiving straight lymph like what the lymph nodes do. The spleen is now associated with after we've combined with the blood. Okay. We know that this is going to remove worn out red blood cells. I don't know how many of you guys know about the spleen and what it does. I have no idea, but yeah, what it does is removes damaged, worn out, old red blood cells. That's what it's supposed to do. It also has a function in the immune system, which is to filter pathogens in a similar way. So we have phagocytes in the form of macrophages that will localize to that tissue in order to deal with that. We already know about this. Associated lymphoid tissue works like a lymph node, but isn't in a lymph node. All right, blood itself. We have a little bit mentioned about this because we talked about this some when we got into ELISA, but there's going to be a difference between plasma and serum. Okay, so in plasma, basically there is some sort of clotting prevention chemical in there so that the actual red blood cells don't clot. So we have over here the unclotted whole blood. It's unclotted. I want to be clear. Unclotted. You see it down there in the bottom. You think, oh, that looks clotted, though. It is not. It's just we've centrifuged it and separated out the red cells from the liquid portion. But it's not clotted. If you were to, you know, do this with it again, it would just mix back up. So this is going to be adding things like heparin and things like that to the tubes, wherever you're drawing tubes from. But anyways, we have the red cells on the bottom with the buffy coat in the middle. That's going to be your white blood cells, and the plasma, which is the liquid portion of that, right? When I'm talking about serum, I'm talking about our red blood cells, letting them actually clot. So you'll send me a tube. There'll be a red top tube or a tiger top tube, and I'll let it sit out. They'll send it to me, and I'll have it sitting out on my desk. And people, you might even walk up to the lab later and see it sitting there. And be like, you haven't run my tube yet, and I'm like, yeah, honey, I'm waiting for it to clot. Because I have to wait for it to clot before I can spin it and then separate that clot from the liquid portion. When you do it that way and let it clot naturally and separate it, the liquid portion is called serum. And that's going to contain like your antibodies and things like that. Okay. All right, white blood cells. These are our leukocytes. Any white blood cell ever of any kind, whether it is part of the innate or the specific immune response, okay, the acquired, fall into this category of white blood cell, aka leukocytes. So when we talk about leukocytosis or we talk about leukopenia, it's all the white blood cells. Okay. We can split them into two groups based on what they look like in a microscope, their morphology, the granulocytes and the agranulocytes. The granulocytes have dark staining granules. Literally, you stain them with the stain and they have these dark spots in them. The agranulocytes don't have that. That's the difference. It's just that staining, okay? But we can separate them based on that. And it's actually a big deal that we do separate them that way. So let's start with looking at our granulocytes. We'll go through what they are and what they do. But number one, we have neutrophils, 2 basophils, 3 eosinophils, and technically 4 mast cells. I don't talk about these a whole lot, but they're going to be involved in allergies mostly. So those are our granulocytes. Okay, our agranulocytes, no granules in these guys. We can divide this into the lymphocytes and the non-lymphocytes. I would say they all precursor from monocytes, and they can become macrophages or like dendritic cells. Okay, so we call them monocytes because they have 1 nucleus. But these are all of the agranulocytes. So the monocytes and their derivatives, and then the lymphocytes. That's it. So the lymphocytes just get the only ones I need you to worry about. I know there's other ones, but I really don't need you to worry about anything except for the T cells and the B cells. I'm aware there's other ones. You don't need to worry about that. Okay, so that's not too bad, right? Monocytes and their derivatives, T cells and B cells, agranulocytes. If this is hard for you somehow to remember which one is which, because I tend not to ask about the mast cells, I will let you know that when I'm talking about granulocytes. I'll ask you which of the following is not a granulocyte, and I'll usually have fill, and then like T cell. Okay, so like neutrophil, basophil, eosinophil, all of those guys are granulocytes. Okay, if it has fill at the end, granulocyte. Easy, easy. So B cells and T cells, the letter cells, lymphocytes, they're agranulocytes. And then the monocytes are kind of by themselves. Mono, think mono, 1, single, they're just by themselves. Over in the agranulocytes, they're kind of just hanging out. But mono, and you can even think about mono being like they have the nucleus glob, but no other granules or anything like that. So it's just 1, mono. We'll come back to the mast cells another time. All right, so that mononuclear phagocyte system, I warned you guys we were going to go through the parts involved in all of this structure setup. So that mononuclear phagocyte system, or the MPS, is just connective tissue fibers. They call it a reticular system. That is just literally just like fibers made of like protein or something like that, that allows cells to move around between cells and between tissues and stuff like that, right? So it's going to connect nearby cells and provides passageway between tissues and organs. And all of our white cells can move along that. All right, so the monocytes, we already talked about the monocytes being agranulocytes, right? So these are the precursors to macrophages, which are going to be our big daddies. These are the ones I'd definitely be familiar with. And then dendritic cells. These are usually associated with the skin. So these monocytes often, and their derivatives, hang out near portals of entry, whether that is, mouth or respiratory area, places like that, waiting to come into contact with invaders. They also have specialized cells, specialized like macrophages. In the liver, we call them Kupffer cells. In the lungs, we call them alveolar macrophages. In the skin, Langerhans cells, and in the brain, microglia. So they have their own macrophages basically associated with each tissue. They get their own name. Now, macrophages in the lungs, those alveolar macrophages, that's where tuberculosis bacteria live. That's where they prefer to live. They want to live inside of those. And I'm telling you right now that this is part of the, innate immune system. These are immune cells. They're phagocytes that those will live inside of. Okay, so cool. All right, that's that. Cytokines, these are just signal molecules, right? So maybe proteins or similar chemicals that will get exuded by a cell to signal the cells next to it, or get pumped into the bloodstream to call for action from other cells, okay? So that's what these are. They're signaling molecules. They are produced by monocytes, macrophages, lymphocytes, fibroblasts, mast cells, platelets, and endothelial cells in the blood vessels. Anything that could be encountering the inflammatory response, basically. So there's 4 categories for these signaling molecules. The pro-inflammatory, to encourage the inflammation, the anti-inflammatory to discourage, it's time to calm down now. The vasodilators and vasoconstrictors to help with blood flow to that area or tell it to die down. And then growth factors, which help regulate, like it says, lymphocyte growth or activation. So these are just chemicals that are going to help with promoting the growth of those acquired immunity cells. So we're not going to go over each one, but there are some examples here we could point out. Things like interleukin, tumor necrosis factor, histamine, right? We know this one quite well. A lot of these are, you've probably heard of some form or another of them, but they usually function in our immune system, especially with the inflammatory response. All right, so physical barriers, we've already mentioned a little bit. You can guess things like tears and mucus and the skin itself and stomach acid are all going to be helpful barriers to protect us. So this isn't going to be news, but the skin also has a waterproofing protein on it called keratin that helps kind of create that sealed barrier, makes it impervious to a lot of things. Very few pathogens, as it says, can penetrate that unbroken layer. The ones that can penetrate the unbroken layer tend to be already like large organisms like worms and stuff like that. Yeah, so remember our outer layer of skin. I mean, I could probably go like this and lose quite a few of my skin cells just from doing that. So your skin cells are constantly sloughing off and they'll take microbes with them as they do that. So that's helpful. Hair follicles, and they shed themselves as well. Skin glands to produce flushing effect of sweat, mucus, trapping things or flushing things out. Saliva with lysozyme, we'll get to lysozyme. Respiratory tract, we have the whole respiratory tree and how it functions. We have a ciliated epithelium. That means we have these little like tiny little finger-like appendages that are constantly moving mucus out of the lung. And so there's mucus in the lung and things go get stuck in it because you breathe in air and there's just stuff. And then the mucus gets moved out by the cilia and coughing helps that. So that's just helping move that out so it can't take hold down in your lungs. We know we have urine and mucus and other fluids in the genitourinary tract that can protect us. Our biome provides microbial antagonism, like we've said, using the nutrients and blocking the living areas, the stadium seats, like I say, blocking access to the epithelial surfaces so that the bad guys can't sit down. Lysozyme, this is an enzyme. So this is a giveaway. We already know that things like ACE are enzymes, right? But there's also enzymes that end in zyme, and isn't that easy because it gives itself away. I like that one. But lysozyme is an enzyme that hydrolyzes peptidoglycan. That just means breaks it down. Okay, so it's going to break down peptidoglycan that's present in the cell walls of bacteria. So any bacteria that have any kind of peptidoglycan, they can break that down. So that's pretty useful, right? Normal, just fight against bacteria. All right, stomach, intestines, and other fluids like semen and the vagina with pH and other antimicrobial chemicals. I'm not going to go into detail right now. Phagocytosis. Okay, the three main types of phagocytes, the cells that are actually going to be the cells that eat up the foreign invaders. Like I keep talking about them, they exist, they do a thing, and they are here. Neutrophils, monocytes, macrophages. Remember, monocytes are going to be in the bloodstream typically, not associated with tissues. Once they go to a tissue, they differentiate into macrophages. So they have similar functions. So neutrophils, monocytes, and macrophages are our main phagocytes. That's important because they're going to be important later on for activating our other systems. The neutrophils react early in the inflammatory response. A lot of times early on in the infection, you'll see high levels of neutrophils, and they're usually the main component of pus. When you have a pus buildup in an infected area, infected wound, typically, they usually are what is comprising it the most. They are also the most numerous white blood cell in human blood by far. So if you do a CBC, we're used to seeing more neutrophils than anything else. Next, we have eosinophils. These are usually associated with parasitic infection. Go figure, we don't have parasites in America, right? So you can go to the doctor and look at your CBC and see that if you have high eosinophils, it probably means you have allergies to something. So that's kind of interesting to think about. All right. Monocytes get transformed into macrophages when they get out of the bloodstream and get into exposed to certain stimuli in the area or whatever. So what happens in phagocytosis? There's steps involved, and of course, I'm going to want you to know them, and I'm going to test you over them. So we have chemotaxis, responding to a chemical in the area. Chemotaxis, we get some sort of protein in the bloodstream, and that draws us to the area, let's say. Ingestion, this is going to be, here it has adhesion of bacteria, but whatever. Engulfment by the thing that you're attaching to. The phagolysosome formation. So you eat up the thing, you form these vesicles, you literally like pinch them off into vesicles inside of the cell. That's the phagosome. Okay, the zome being the vesicle, phago being what you made from eating the thing. Okay, so you've got your phagosome. Lysosomes are vesicles containing chemicals that are going to kill and break down the bad stuff in there. Okay, so those will merge together. When they do that, they become the phagolysosomes. Yeah, go figure that lysosomes contain lysozyme. Yeah, they do. Okay, they also have those toxic oxygen products and other things like that. Anyways, that will lead to destruction of the microbe. And then after we've destroyed it and broken it down, we can excrete the parts. You can use the parts as part of your cell, but a lot of times they'll just pop it back out. Okay. All right, so chemotaxis and ingestion usually is going to be signaled by the presence of pathogen-associated molecular patterns, PAMPs again, right? These are signal molecules on the surface of the microbes. Things that are great examples of PAMPs, peptidoglycan, right? Our cells don't have that. So that's just, if you have it at all, bad guy. Lipopolysaccharide, that is, remember what this one is? Endotoxin on the surface of gram-negative bacteria, right? So that's not supposed to be in our bloodstream and stuff like that. So that's immediately bad guy. And if we have double-stranded RNA, none of anything in our process in our body naturally has double-stranded RNA. So that's also a signal that that shouldn't be there. So that's a good one too. Those are PAMPs. So it's just immediate bad guy signs. And they're recognized by these pattern recognition receptors that we're not going to go over. All right, the phagolysosome. The phagosome itself formed whenever we eat the bad guy. The lysosome containing the chemicals comes and merge with it. That's the phagolysosome. Kills bacteria typically within 30 minutes. It also has oxygen, toxic oxygen molecules in there. I mean, I'm not going to test you over the exact, what is going on in there, but there's other stuff like lactic acid, lysozyme, nitric oxide that can all be involved in killing the bad guys. All right, moving on to inflammation. Signs of inflammation, these 4 terms, I would be aware of the 4 terms. There's like a question of like, which of the following is not one of these? And it's going to be some made up word that looks quite a lot like these. So be able to pick it out, okay? Rubor is redness. Tumor is swelling. That's tumor, swelling. Dolor is pain, and then calor is heat. If you think of calories, I don't know if you guys know this, but the word calorie has to do with the amount of heat energy released by like burning a sugar, like a gram of sugar or something like that. So calor makes sense to go with heat. That one's a pretty easy one. But we've got tumor for swelling. Rubor also starts with R. So then the only one that's left is dolor. You can remember it. I always forget dolor, like always forget it. All right. So those are the 4. There's also a 5th one, which is just loss of function of the area. Yeah. So it doesn't get its own little word, but there it is. So if you have loss of function area as a response to inflammation. All right. Chronic inflammation, you have all this going on for a long period of time and all these signals to bring inflammation into the area. This is usually a feature of autoimmune diseases and chronic diseases as well. We see this with long COVID, and it can affect entire systems, blood vessels, lungs, skin, and joints over time because following that, all that inflammatory response into the area. So what is the point of inflammation? What does it do? Why do we have it? What's happening here? So it helps to mobilize and attract immune components to the site. So anything relating to fighting these guys that isn't acquired because we don't have that going yet. But anything else related to this, we're going to mobilize it to the area. It's going to set in motion mechanisms to repair tissue damage in that area, as well as clear away any harmful substances. And then we're going to hopefully destroy some microbes and block invasion. So this is really what they're good for, blocking further invasion. They're going to do what they can to destroy the microbes while they're doing this. But mostly they're going to be called to the area and also exude their own chemicals that'll be calling other cells to the area, activating our lymphocytes and, having them proliferate and everything like that so we can get them working. All right, so here's how this would work. You might have an injury where at first you might have vasoconstriction because you're trying to prevent things from getting into the body. So you'll constrict that area, but eventually you'll have vasodilation to help the cells and everything move to the area. Anyways, so vasodilation here, once that's ready to move things like neutrophils and stuff into the area, we'll start forming a clot, which you know how clots work. It's going to be things like platelets and everything like that building up in that area, drying up and whatever. So then we have number 3, we have edema and pus. Edema, just bringing that fluid into that area, and as it builds up in that area, has all of those signal molecules and everything that we need to have our neutrophils go to work and do their work, right? And if that's happening, ideally how it should, then pus will form because there's a lot of neutrophils and dead bacteria and stuff in that area. And so eventually you cleared everything out, you've fought everything off, and all of that fluid can move out again of the area back into the bloodstream, and everything can heal up with new tissue formation and all of that. Our lymphocytes have come into the area and helped to kill off any infected cells or bring antibodies or whatever it is to help prevent that infection from spreading and all that. And then you might have a scar there because your body doesn't care what you look like. All right. So earliest changes will be in the vasculature. Then we'll have other changes controlled by the cytokines, typically, and other chemicals that are going to be released by blood cells, tissues, platelets, any tissue in that area is literally, any tissue in the area is capable of producing some sort of chemical or signal that can affect the inflammatory response to some degree. And based on its exposure to whatever's going on, can produce certain signals to either help it or dampen it or whatever it is. So, yeah. So yeah, you want inflammation because like the redness just happens because the blood flow there, right? The tumor, the swelling happens because of the fluids flowing in with the cells that are coming to fight. The dolor, the pain, is literally usually just because of the pressure in the area, putting pressure on the nerves itself or damage literally to the nerves. That some chemicals can activate pain as well, simply to let your body know, like, hey, something bad's going on over here. And then, what was the last one? I can't remember what it is. I already did redness. Now I gotta go back to it because now I feel stupid. Yeah, the heat. Okay, well, yeah, I feel like the heat makes sense though, because you're bringing the blood to the area. So that's the redness and the heat come with each other. All right, so anyways, that's why we get the actual visual aspect of the inflammation going on. It is your body just trying to send cells and chemicals to the area to help out. Jeez, man. All right, exudate is anything dealing with escaping that extracellular space. So any leaking of fluid. Edema is swelling due to fluid in the area. Diapedesis is an important term that will definitely be on the test as well. It is just movement of any of these white cells, any of the white cells, from the bloodstream into the tissue. They just squeeze in between these cells here of the epithelium, endothelium, and then move into whatever tissue they're being signaled to move into. So that's that process of getting out of the bloodstream and into the tissue. That's diapedesis. They change their shape and literally just squeeze through. And then move by chemotaxis. We kind of already know this. It's going to be chemical signals, and they'll move based on those signals, as will the fluid itself. All right. Later on in inflammation, we will have arrival of the B cells and the T cells. And that, like I said, it's basically the end of the fight, man. You brought in the tanks now, and we don't have to keep fighting with our, puny little guns or whatever. So the B cells and T cells serve in that. All right, fevers. Fevers are part of inflammation, or at least related very closely to it. Here we have an abnormally elevated body temperature. Calor, right, is what we had with inflammation. So that happens with our whole body. So abnormally elevated body temperature, it's a universal symptom of infection, It's associated with also with allergies, cancer, and other organic illnesses outside of that, but we're talking about the infection aspect of it. Ranges of fever. I'm sure you guys don't need me to go over these because I'm not going to test you about the range itself, but there are low, moderate, and high fevers. A pyrogen is anything that activates a fever. So it could just be the presence of certain bad guy molecules that can trigger a fever, or it could be those cytokines, those signaling molecules that are produced that can trigger the fever. So if it comes from the bad guy itself to trigger it, that's exogenous, right? Endogenous, our body is producing the chemical that causes it. What does fever do for us? What's the benefit of a fever? It inhibits multiplication of temperature-sensitive microorganisms, which a lot of them are, surprisingly. It impedes the nutrition of bacteria by reducing the availability of iron. It involves production of like lactoferrin or something that like basically hoards the iron away so the bacteria can't have it and they need it for growth. It increases metabolism and it stimulates immune reactions and naturally protective physiological processes. You can imagine like we're talking about warming something up, that our cells could actually be activated by that and be more active as a result of that warmth. That's exactly what's going on here. We're speeding up hematopoiesis, which is just generating new cells. Phagocytosis and the adaptive immune response, also sped up by the heat. Okay, cool. So do you treat fever or not? Slight to moderate, you might let it run its course. Of course, this is up to you as a parent. Use your judgment and of course call your doctor where necessary, but high and prolonged fevers should probably be treated. And if you have any underlying conditions outside of that situation as well, then you might want to watch it more closely. So interferon, we've already mentioned interferon as being that chemical that's like the guy sees somebody breaking into his house, he goes and hides, he calls his neighbor so the neighbor can get his shotgun out and protect his family before the bad guy gets over there, right? The burglar, that's interferon. So it's involved in all this immune process, obviously. So here it is, here's just a picture of what I just explained. Here's a virus. We activate our interferon gene. The cell says, no, 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 this is bad, this is bad. Interferon gets made, moved, signaled to the next cell so that whenever this virus comes in, it's already got antiviral proteins ready to go in its shotgun. So let's see. And we can use this to treat certain infections. We can take interferon like made like with biomechanics or whatever, bioengineering, and use that to help people fight infections, you know, control that. So it's useful that way. All right, we have complement. I think this is the last bit of it. Complement. It is 50 blood proteins. Now I added this part in because it wasn't in here, but you should probably know it's always in your blood. This part, your complement proteins, it doesn't have to be induced. They are always there in your bloodstream constantly, 24/7, 365, always there, okay? These 50 proteins, they work together to destroy bacteria. They have a cascade reaction where like one thing happens and that triggers the next thing and the next thing and so on and so forth. It's a sequential response. So it can be activated by antibody, or there's three ways, okay? The first one is antibody. The second is the presence of sugars on the microbes. Sugars on microbial surfaces that are recognized by lectins. So we call it the lectin pathway, okay? But we're looking for sugars on the microbes. And then that we don't have, it's usually mannose. 3 is the alternative pathway. So this is repeating molecules on microbial surface, and that's usually related to things like LPS or maybe peptidoglycan, something like that. That's just a general repeated molecule on top of, you know, what's there as the microbe. So 3 complement pathways. Antibody triggers it, lectin binding to sugars triggers it, or the alternative is LPS and peptidoglycan. All right, so these are the pathways, no matter what, no matter how you trigger it, either of these, this whole cascade of C3 becomes C3A and C3B. C3B induces phagocytosis. C3A is involved in triggering inflammation. And you can even take C3B even further down into the membrane attack complex. And I'll show you what that is in a moment. And whatever. And those proteins then, C6 through 9, will build the membrane attack complex. So we have three main functions of complement here, right? We have, it helps with phagocytosis, it helps with inflammation, or it creates the membrane attack complex. It's literally, we would abbreviate as MAC, okay? All right, the 4 stages of complement initiation will be just literally just initiation. Something interacted with our complement proteins that were always, floating around, the 1, 2, or 3 pathway. That gets amplified, and then we have development of the cascade. They will then polymerize. They usually bind with one another and then form the membrane attack complex. Literally, the proteins come together and form a hole in the membrane. So we're going to talk about forming holes in membranes before with our chemicals, but this is a little bit different than that. These are designed to form holes as proteins produced by your body in, these invaders. It's pretty cool. That's the membrane attack complex from complement. Okay, that was everything. Yes. I do want to say, we'll get into it next time, whatever. So here's your thing for your picture. Are monocytes important for the innate immune response, or might they play any other important roles? Well, I'll tell you this, we didn't go over it much in this chapter, but monocytes, because they are phagocytes, and as well as macrophages and neutrophils, they break down the bad guys, right? And take the molecules of bad guys that say this is a bad guy, and then go and present them to what we're going to learn about in the next chapter, our lymphocytes, to try to activate them. And they say, hey, do you recognize this? And then hold it out. And if it does recognize it, then it activates that pathway. So they're important in both sides, the innate and the acquired.